Good evening to one and all present here. I, Amina Dholkawala, on behalf of the Department of Microbiology, KC College, extend a very warm welcome to everyone present today. We appreciate you joining for today's session, which is Microbe, Biome to Application, and hope you all will learn a great deal today. To begin this program, I would first like to call upon Dr. Sejal Rathod, HOD, Department of Microbiology, Coordinator, Department of Biotechnology, to please deliver the welcome address. Over to you, ma'am.
Thank you, Amina. Yes, so very good evening to everyone, all the students of Microbiology, Biotechnology, and the other departments that have joined us today. I welcome you all to this program that we have organized under CBT Star status. That is a lecture on microbiome to application by Dr. Anjali Bose, who is the assistant manager at Zytex Biotech Private Limited. We at KC and at the HSNC University have always strived to make a balance between academics and at the same time, making the students aware of what actually is happening in research institutes, in industries. And so in this direction, towards this direction, we keep on organizing such a series of lectures for all our students where, we invite people from research institutions in India, abroad, and of course, industries, so that they are well aware of what is happening in those institutions. So learning something in the class is different than what is actually applicable to the real world outside, whether it's research institutions or industries. So to meet this, or to bridge this gap, we are having one such wonderful session today by Dr. Anjali Bose. So welcome, ma'am, to this STAR DBT status session. And I request you, okay, Amina, over to you. Let's have a very enriching and informative session. Looking forward to it, madam.
Thank you, ma'am. I would now like to call upon Mrs. Rajita Satish to please introduce our speaker for the day. Over to you, ma'am.
Good evening, everyone. It's my pleasure to introduce to you Dr. Anjali Bose, who is currently working as Assistant Manager in the Strain Management Department of Zytex Biotech Private Limited, Vadodara. Ma'am has completed her PhD in microbiology from Sardar Patel University, Gujarat. She has also completed several other courses, including a professional diploma in software engineering. Ma'am has also worked in University of Hyderabad as postdoctoral fellow. She has worked as a research associate in Sardar Patel Renewable Energy Research Institute, Anand. Ma'am is a recipient of several awards such as UGC DS Kotari Postdoctoral Fellowship 2013, Research Fellowship for Meritorious Student by UGC 2009, to name a few. Ma'am has several publications in peer-reviewed national and international journals. To name a few: Annals of Microbiology, Microbiology Resource Announcement, Applied Microbiology, Biotechnology. Ma'am is a member of several organizations across India and reviewer of national international journals, also an active runner in several marathons. In Zytex Biotech Limited, she has established a fully functional lab for strain and process development, established culture banks, performed probiogenomic and pathogenomic analysis for commercial strains. May I now request Dr. Anjali Bose, who loves to introduce herself as a biotechnologist with a rich experience in applied microbiology and fermentation technology, to deliver the talk on microbe biome to application.
Should I start now?
Yes, ma'am.
Okay. Hello, everyone. Good afternoon. I hope everybody will enjoy this presentation. Can you put into presentation mode? Yes. So this is actually a very good opportunity where I can actually communicate with the students who are planning to decide their career, maybe in industry or PhD. So this presentation will actually help them to have an insight how fermentation companies or industries work. So I have kept this very simple as microbe, biome to application. Next, please. So before starting with my presentation flow, I would first like to tell you what exactly biome is. So biome is a region or a sphere of earth which actually have a defined climatic composition which harbors soil, flora, fauna, microorganisms which are limited to particular region and adapted themselves to that particular climate. In short, many individual ecosystem coexist to bring upon a biome. If you closely look into this biome, there will be crores or trillions of microorganisms which are present as per the defining environment, but they have many genes which can be actually exploited for betterment of human mankind. So this whole process of exploring new biological material from different climatic region for commercial application is termed as bioprospecting. Now, this is a very fascinating term which is coming in play these days. And to define how bioprospecting actually happens, these are the 6 topics which we will go through this presentation. The first step of this bioprospecting is isolation screening, then preservation, then how we decide or define a media for taking up a small microorganism to a bulk product stage, the sterilization which is involved for the production procedure, then how it is being scaled up, and finally the upstream and downstream of the product. Next, please. So you must be aware of the different isolation and screening techniques. So I don't want to go deep in that. The major questions which I mostly ask in my interview and people do not able to answer these questions are, why do we want to isolate these strains? So the simple answer to this is, we want to culture them in lab. Then the next question is, why do we want to culture them in lab? The thing is, if we do not culture the organism in lab, how do we characterize them? So characterize means what are the different properties it has, how we can exploit it for the betterment of humankind. So to know all these, we need to culture them or preserve them in lab and regularly analyze them for our own betterment. So that is the reason why we want to isolate them. The next culture is, can't we work with the mixed culture, or why not mixed culture? So the thing is, if there is a sample given to you, say a soil sample or a water sample or a sludge sample, there can be so many number of microorganisms lying in it. But the thing is, if you don't know which all microorganisms are doing what all activities, you cannot exploit that particular organism for your use. So, indeed, we can work with a consortia of culture. Like, we have to first isolate a particular organism, characterize it, see how it can be applied commercially, and then mix them finally to use in a particular proportion before being used in environment. For example, biofertilizer. Everybody knows these days organic farming and biofertilizer pesticides are in hype. So earlier what farmer was doing, he was applying urea in the field. What happens if you do not apply a particular amount of urea, the plant will die. There are other side effects like they are spraying pesticides which is actually being ingested by us when we eat the vegetables. So the different aspect to look this problem is, if we produce something which is biodegradable, anything which is biodegradable is of biological origin. Now, any single organism cannot provide all the different application. Say it cannot control the pest at the same time when it is providing nutrition to the plant. So what these intelligent people do, they find out different microorganism, one which is responsible for protecting the plant from any disease, the other organism which is responsible for the intake of mineral, and third organism which is responsible for producing some phytohormones which actually responsible for the growth, increment of the canopy, that is the plant body. So then they mix up, then they characterize. First in the lab, they characterize all these organisms individual, and then they mix up in certain amount, and final product is then marketed, which the farmer can happily add. So yes, we can produce a product which is having a mixed culture, but we cannot work or characterize a mixed culture. Last, then can we work with unculturable? So yes, we can work with unculturable. Now the word unculturable means whenever most of the audience I believe is a microbiologist. So whenever I asked what is a microbe, it is bacteria, fungus or virus. So mostly we cultivate bacteria or fungus or streptomyces in lab. So if something which we can culture or sub or like streak or spread, or we can see on our culture media on the tabletops, those organisms can be marked as culturable. But there are lakhs of other microorganisms due to lack of environmental condition. We are unable to mimic the exact environmental condition required in which they actually grow, and we are not able to provide them the sufficient nutrients which are required for their growth, which are actually essential for their growth. So these organisms cannot be cultured in lab, but they do exist, and many a times they have many interesting genes which can be exploited and commercialized. So what people do, they take the sample, say soil sample, they isolate the whole genome, like the total genome of that soil sample. Then they try to fish out a particular gene from it, they clone it, and then they use it for production if that gene seems to have some commercial application. But in general, unculturable bacteria could not be worked upon. Only the way out is by cloning their genes and using them commercially. Now, mostly the industries prefer wild strain. Why don't they prefer GMO? You won't believe, we are dealing with customers abroad, mostly in Europe and USA. Although every biotechnologist is fascinated to use genetically modified bacteria or plant, because we learn all those things in our coursework. But the thing is, when any organism is genetically modified, it gets prone to spontaneous mutation. Now, if mutation keeps on happening, then there can be chances of uh, changing the genome in such a manner that it can become some pathogen. It can have some pathogenic trait and can be harmful to human beings or animal or plants to whom it is applied to. So to limit this, mostly commercialization is being done with the wild strain, because the chances of mutation in these wild strains are 0. Mind it, 0.0001%. That is, the clones or the subcultures are 99.999% same, genetically same to their parent culture. So that is why mostly wild cultures are being used. Now, before going to say I want to set up a commercial entity, so before going into that, I should first decide what is my objective to take up this isolation procedure. I need some objective to be defined at the initial goal. Say I want to produce protease enzyme, then I have to define my objective. I want to produce an enzyme. Which enzyme? Do I want to go for diversity study? Do I want to check what all different population coexist in a particular sample? I will give you an example. When I was doing PhD, I was approached by an industry which was producing heavy water. The problem was the heavy water which was being produced was getting fouled. It was having some foul smell. So now you must be aware this heavy water is being used for atomic energy generation. But if it is having some foul smell, then it is not advisable to use. There must be something going on. So they asked us to isolate those organisms. So it always depends what type of objective I have. Depending upon that, I have to design my strategy, like from where I have to isolate the organism. Say I want to isolate lipase producing organism and I get some soil source from my house. Will that be efficient enough to search or dig into those organisms which are lipase producer? No. So I have to search for a sample which is rich in oil. Why? Because if a sample is rich in oil, it will definitely have some organisms which are producing lipase to consume these lipids and utilize it for their energy purpose. The chances of getting such organism will be much higher in a oil contaminated soil. So I have to first design my strategy how I have to choose. Then second thing is I have done the sampling. Then how I will take it to the lab. Say a culture, I have done sampling from Antarctica. Now the organisms growing there will be psychrophilic, that is they are growing at low temperature. If I transport them in normal condition, I won't be able to culture them in lab. By the time I reach the lab, they will die. So these are things, minute things I have to keep in mind, and I have to prepare a strategy while doing before the isolation procedure. Now these are the different isolation techniques, majorly streak plate, pour plate, spread plate, micropanipulator, and I'm very sure that your teacher must have taught you these techniques in detail. I just only want to say, do you really know when and why these different techniques are used? So I'm not sure, but let me explain that. So mostly we use streak plate whenever we want to check or see the purity of the culture. So if you want to check the purity of the culture, you will not use pour plate, you will not use spread plate, you will only use streak plate technique. Okay. Then second thing is when you are trying to, when you go for pour plate or spread plate, that is anything which is to be checked for pour plate or spread plate need to be diluted, serially diluted. Why? Because any sample will have lakhs and crores of organism in it, and if I directly pour it, I will get mixed culture growth, which will be a lawn or a mat growth, and I won't be able to isolate them. So it's better to serially dilute them and to reach a level where I can pick or enumerate them. So pour plate is used when I want to look into the total diversity of the sample, when I don't want to miss even the microaerophilic and aerophilic culture. So I can achieve this. If I want to see the total diversity, I can spread it also. But the thing is, the cultures which are not able to grow in presence of or excess of oxygen won't be able to grow in such plate. So pour plate methods are much better when we want to look into the diversity. Then we use serial dilution along with pour plate, where the embedded colonies will be the colonies which are microaerophilic or maybe sometime anaerobic. In spread plate, mostly this technique is used when we want to enumerate. That is, we want to count a sample. How many cultures are there in a particular sample? Then we use this spread plate method. That too only for the aerobic or slightly microaerophilic culture. Now, micromanipulator is a cost-intensive procedure. It is, I have kept a picture along with this. So there is a capillary with a piston, and we have a slide in microscopic arrange in a adjusted under the microscope, and in a hanging drop, while looking through the microscope, we can pick a single culture, a single bacterial cell or fungal cell, and later on, microscope assisted only, we can transfer it into suitable media. So this is a very cost intensive method. And second thing is we need trained operators. to deal with it. But the best part is we are very much sure that we are working with only single cell, and we will get a pure culture from this type of streaking or this type of isolation technique. Now, screening and isolation sometimes go side by side. So screening techniques are majorly divided in four types: enrichment, selection, differentiation, and quantification. Now, keeping the same example, if I want to work with protease producer, then I want to get a sample which will have more, likely would have more number of proteases producing organism. Secondly, while culturing, I want to increase their population. So I will add something in the media which will enable those organisms to fastly grow and overpower others. So this process is known as enrichment. Second is selection, where I want to select a particular group of organisms and I do not want other organisms to come into play. So I can use some antibiotic. Third is differentiation screening, where I want to differentiate. Like if I want to work with lactic acid producing bacteria, okay, or organic acid producing bacteria. So what I will do, in a simple media, I may add calcium carbonate. So calcium carbonate is a powder-like substance which is majorly used in talcum powder. Because I will add this, it is insoluble, it will make the whole plate chalky, that is white colored. Now, whenever the organism will grow, it will produce lactic acid. Those organisms which are producing lactic acid, the media pH will drop because it is an organic acid producing bacteria. And because the media pH drops, the calcium carbonate get disintegrated and solubilized, thereby resulting is a halo zone. And we can then differentiate that these are the lactic acid producing bacteria and which are not producing halozones are normal bacteria. Then quantification. So say I have 5 different lactic acid producing bacteria. Now I want to know which one I have to go with for final commercialization. So I need to quantify how much amount of lactic acid it is producing. Which one is the fast producer? So I need to quantitate the amount it is producing. So that can be done by colorimetric method or spectrophotometric method. So that is a screening technique which is being used. Next, please. Yeah. So second is preservation. Now, why do we want to preserve? Because mostly what happens, if I regularly subculture it in lab, then the viability of the culture may be lost. That is, if I subculture it, I will use agar plates and the plate may get desiccated and I may not be able to re-isolate the culture from the plate. So I need to maintain a viability. Second, I need to maintain its purity. While course of work, it should not get cross-contaminated with other cultures which are being worked upon in the lab. Third is, I also want to restrict the genetic changes which may happen during the subculturing. As I told you, that there is a stance of spontaneous mutation. So one base, one for every 1,000 base has a chance of going into mutation. And a genome contains 10 to the power 6 to 7 base pairs. So just imagine the amount of mutation can happen in one subculturing. So that is why I do not want to subculture it again and make it prone to mutation. So these are the three fundamental things why we want to preserve a culture. I have listed few commonly used preservation technique. One is subculturing, that is when we want to maintain a live copy, when we want to regularly use it. At ultra low temperature, that is at refrigerators, in refrigerators at 4 degrees, just click once. Yes. And then I can also preserve it in deep freezer, that is from minus 40 to minus 80. I can keep it in liquid nitrogen and I can also preserve it under nitrogen vapor. As you must know that liquid nitrogen has a very low temperature. So at such a low temperature, it is definitely the water content inside the cell may get freezed. If a water freeze, it forms ice crystals. Now, if they form ice crystal and we thaw them, take it out at normal temperature, these ice crystals will melt down, leaving behind pores in the cells, which can cause lysis of the cell and actually cell death. So when we are preserving at a low temperature, we need to add cryoprotective agents, which actually limits these ice crystal formation. Okay, so there are several cryoprotective agents which are being used, like glycerol, DMSO, bovine serum, albumin, skimmed milks. So what happens? These molecules like glycerol molecule or skim milk molecule, the protein molecules, they form a coat around the bacteria, and when the ice crystal are formed, these crystals could not able to pierce through the cell, and that is how the cell is maintained at such lower temperature without any injury. There are many fungus which are not able to, we cannot store them at so low temperature because they actually die. We cannot store them. So for those purpose, we use mineral oil overlay. So the culture is treated on agar slope and a paraffin oil is filled at the top so that the oxygen cannot enter and metabolic activity will be restricted, and that is how these slopes can be kept for around 5 to 10 years. Then there are few other examples, like sometimes fungal spores and some endospore forming bacteria. They can be stored in sterile soil. So they are mixed with sterile soil, kept for a few days to grow in and achieve their vegetative growth, and then they are stored in normal refrigeration condition. And many a time it has been found that they can survive up to 10 to 20 years. Then the best method, which is being used in every industry, almost every industry, be it single cell protein, be it culture, bacteria, be it fungus, is lyophilization. So what happens is, in lyophilization, mostly when we freeze something, uh, there is a change in state or the phase of water. Okay, so water will first freeze, then it will liquefy, and then it can dry. But here what happens? We first freeze the cells at minus 70 degrees, then under vacuum, it is being dried. The water do not melt down, it directly sublimate and evaporate to dryness, leaving behind only the cells. So, so what happens? It do not thaw, because of which there is no injury caused to the cell. And such kind of, and because vacuum is maintained, there will be no metabolic activity going on. And such lyophilized cell can be stored for around 70, 80, or maybe sometime 100 years. So most of the culture collection banks, they are preserving their culture either in form of lyophilized stock or glycerol stocks that are in liquid nitrogen. Next, please. Yeah. Then then we will be discussing the most critical part, that is the media. So why media selection is very critical. So I have summarized so that if somebody wants to just go through it, you can quickly get into a picture in your mind why this media selection of media component is very critical. This is because, say, I want to produce protease. Again, the same example, but the organism is having an ability to produce lipase or cellulose along with protease. Now, my aim is to produce only protease. So, I will have to design the media and select components in such a manner that it will only produce protease, and the other byproducts or other enzymes which the organism is capable to will be limited to a very lower extent. So it will maximize product yield, it will minimize undesired byproduct yield. Second, say the organism is producing protease. Protease is a primary metabolite. It will be produced in exponential phase. So it is producing maximum protease at 48 hours. But I want to speed up the reaction, so I will select such a media so that my organism is able to produce it in 12 hours. So what will happen? See, when there is commercialization of something, money is involved. So if I'm taking 48 hours to produce something, and if I could reduce it to 12 hours, I'm saving time. It means my fermenter is free for another run. So I will be earning money where I was able to take only 3 batches in a week. Now I am able to take 6 batches in a week. So I have to design a media in such a manner that it could increase the rate of product formation. Then there are two important points, that is ease of downstreaming and ease of sterilization. I think everybody of you have seen your mother cooking rice. So have you ever observed that the starch which is drained off is so thick? Same is happening in the fermentation broth. Many a times, if we select starch during the sterilization process, it becomes thick. And due to the increased viscosity, the sterilization process can be hindered. So either we have, if we want proper sterilization, we have to increase the time. So while selecting, we have to think which media will be fast, how easily it can be sterilized. There are many components which are insoluble. Although the organism can produce enzymes which can solubilize these organisms, but if something left undissolved, then it can hinder during downstream processing. So these are the few tips which we should keep in mind while media designing. Second is different types. So you must be aware, depending upon gelling, there can be solid, semi-solid or liquid. So this is the type which we mostly use in fermentation at commercial level. The solid media is mostly used to check the purity or sometime also for solid state fermentation, like vinegar production. So Aspergillus niger is thrown over the hay, which are kept in trays for several days, and then these trays are harvested, they are squeezed, and the amount which is coming out will be vinegar, which will be then purified. So there can be solid media, there will be semi-liquid media, which are majorly used for transportation. So when you want to transport one culture from some place to other place, then mostly we use semi-solid media, sometimes to also check the motility of the culture. And liquid media compromises about 90% of the fermentation industries, where the media components are dissolved in water, which we known as which is called as fermentation broth, in which the culture is grown. Then nutrient can be complex and synthetic. We will discuss this later. And then function, enrichment, selection, differentiation. I have already discussed during screening, and you must be aware in detail about the different media like McConkie and chocolate blood agar. So I won't go in deep with this attribute. Then the major thing, like in human body, around 80% is water. The earth is around 70 to 80% of water. Similarly, in fermentation, water comprises of the major portion of the process. That is, in media, water is required. For sterilization and cooling, water is required. Why? To generate steam, which is actually required for the sterilization process. Then chilled water is required for the cooling process. Then before making media, you need to clean the vessel, the pipes, etc. For that, you again need water. Then for downstreaming, for cleaning your product, finally extracting the cell or the metabolite, at every stage you need water. So water is the major component. So if it is so important, then we have to also look into the properties. So major properties of water is pH. Normally it is kept neutral unless and until it is required to precipitate out few things. We always keep the water pH neutral. Second is dissolved salts. So it is a very critical part. Dissolved solids means this portion, RO, we drink water, and these days the water is having a very high TDS amount. Same is with the industrial water. But depending upon our application, say if a company is trying to manufacture vaccine, it cannot have a TDS even 1. So we need a TDS of 0, total dissolved solid of 0. In case of fermentation industries which are producing bacterial, fungal biomass or enzyme, we can have a TDS ranging from 50 to 200. So depending upon our requirement, TDS can be fluctuate. A very interesting example I will give. This water sometimes result, the property of the water or the dissolved solid or mineral content may also result in differentiation of the product. For example, in brewing industry, if there is impurity of calcium sulfate, then the beer which is formed is known as English beer because of the bitterness. If there is a carbonate, then the color of the beer may change to dark. So these are the different attributes. A small amount of mineral content, up or low, may cause such type of changes. Then energy sources, this we will discuss in coming slides. Growth, buffer, precursor and oxygen requirement. Next please. Yeah. So most of the commercially used microorganisms, they either use light or they use the media components to withdraw their energy. So for production of single cell protein like spirulina, anabina or some biomass, there are many biofuels which are produced by, from algae. And how we have to grow this algae commercially, we have to expose them to sunlight so that they can withdraw their energy. They are autotrophic and withdraw their energy from sunlight. So we have big flotation baths where these algae can grow. Or we can also grow microbes in a closed environment where we can provide them suitable media nutrients. From there, they can consume their energy. So mostly the organisms which are used in fermentation are chemoorganotopes. That is, they are utilizing the chemical bonds, they are breaking the chemical bonds of the organic compounds to get energy. And the two major components of media which are responsible to provide them this energy are carbon source and nitrogen source. So coming first to carbon source. If you remember the growth curve, there is a lag phase, exponential, stationary and growth. So in the exponential phase, the culture reproduce and increase in population. So the major products which are a fermentation company produce maybe either bacterial biomass or secondary or primary metabolite. I'm sorry, secondary or primary metabolites, or it can also cause some biotransformation. It can produce some enzyme which will transform one product to another. So for growth, that is biomass and primary metabolite formation, carbon source is directly proportional. If you give more carbon source, the organism will grow more in number and you can harvest greater amount of microorganisms. If you are giving more carbon sources, then more number of, then it will be in exponential growth. So primary metabolites will be produced at a huge amount. But the secondary metabolite production is inversely proportional to carbon source. Why? Because when a culture enters into stationary phase, that is the time when it competes with each other and it produces many metabolites which actually inhibit the growth of other. So these secondary metabolites will only be produced when there is nutrient depletion. So carbon source is inversely proportional to metabolite production. Now factors which are responsible for affecting such choices, that is product expression, purity and quality. Now if I want to, I have to first decide which kind of metabolite or biomass I want to produce. If I want to produce primary metabolite, which kind of carbohydrate I have to take. So I have to decide depending upon my final product, its purity. Say if I am producing vaccine, I cannot use molasses because it will have so many number of micronutrient and contaminants, and vaccines are actually injected in blood. So purification will take lot of time. So it depends upon the purity of the final product, which type of carbon source I have to decide. Then again, sterilization and downstreaming. If the sterilization, we have to always keep in mind that there should be easy sterilization and downstreaming procedure. I have already discussed, it should not be insoluble. Then mineral contents and carbon sources are always accompanied with trace amount of minerals and minerals, that is metal ions. So the ash content of any carbon source is actually the mineral content of that carbon source, and it is essential sometimes for the growth of the organism. More majorly, these carbon sources, while sterilizing, that is while autoclaving, they turn brown. This is because of their charring, and this happens because when they react with the reducing sugar react with the protein part, and they form colorful black colored substances which actually inhibit the growth of the microorganism, and this reaction is known as Maillard reaction. So we do not want to end up inhibiting the growth of the culture. So it is advisable always to separately sterilize carbon sources. Then I have already mentioned that gelatinization. The starch majorly results in gelatinization. Then just imagine a person who is lactose intolerant. So they cannot intake milk because they start vomiting or indigestion problem. So if I produce a final product which has been grown on a media containing lactose, what will happen if even a single molecule of lactose goes into the final product and the person having lactose intolerant consume that, they will show the symptoms. Many people also have chitinase allergy. Now, chitinolytic substances can cause allergy, and if it goes into the final product, it may hamper the consumer. Similarly, gluten allergy. So many people have gluten allergy. So we have, while deciding which carbon source we have to select, we have to keep this in mind. So these are the few examples: starch, dextrin. So starch, when hydrolyzed, may give dextrin, and upon further hydrolysis, give glucose, etc. Fat, oil. These can be used as carbon sources. Coming to nitrogen sources, the factors are, if we know that the organism is producing certain enzymes, then we can use a particular nitrogen source. Say, if my organism do not produce urease and I am using urea as a nitrogen source, will it be able to consume it? No. If it don't has nitrate reductase, it won't be able to reduce the nitrates which we are giving. So we have to check for the first characterize our organism, whether our organism is able to consume it or not. Second, ease of degradation. If I am providing so much amount of nitrogen which the organism is not able to consume, then it will be left in the media, which will cause toxicity of the media. We don't want to end up in that. Then ease of degradation, and then again sterilization and downstream. Now with nitrogen sources, when you try to sterilize it, it has a tendency to form foam. If it forms foams, then the penetration of steam into the media will get restricted. So while sterilizing any nitrogen source in liquid media, we usually add anti-foam, which are surface active agent, which form a particular bridge between the liquid air interface and enables the steam to penetrate through and make perform proper sterilization. So that you should always keep in mind. Then there are different type of nitrogen sources, complex sources, and synthetic or simple sources. Then pH control. Majorly, nitrogen sources are responsible for increasing the pH of the fermentation broth. This is why, because when nitrogen is being used, ammonium ions are being produced. So ammonium ion will lead to increase in or alkalinization of the media, and because of such sharp increase or decrease in pH, the organism may not be able to withstand, and they may undergo lysis or degeneration. So we have to always check whether there is a higher extent of pH. We have to control it, whether there is foaming or not. Again, allergen free. Many people have soya bean allergy. Many people have meat allergy. Many people have fish allergy. So we have to keep in all those things in mind, in which market these products are being sold off. So depending upon that, we have to select. Then for nitrogen sources, majorly all the nitrogen sources are very hygroscopic. They absorb moisture from the environment. So we have to keep in mind that they should be stored in temperature controlled dry places, and so that they do not capture moisture. These are few of the nitrogen sources like seed meals, yeast extract, peptones, CSL. CSL is corn steep liquor. This is a byproduct of corn processing units, and it is both carbon and nitrogen source, but majorly considered as a nitrogen source. Next, please. Now, growth factors. So I won't go much detail in this. Just to touch upon, there are many impurities in carbon source like metal ions, metal ions, which are actually responsible for the growth of the organism. Few examples like calcium, magnesium, they are required for sporulation. Just to make you, you should not go into sleep. So I will tell you. Calcium is actually required for the spore coat formation. So if we give more calcium, then the spore coat formation, the rate of spore coat formation will increase. So we are basically a bacillus forming company, bacillus fermenting company. So what we do, we incorporate calcium sulfate always in our media so that we can get quick sporulation. Then manganese, iron, zinc, these are responsible for secondary metabolite. Why these are responsible? These are toxic metals. So when there is some toxic element present in the media, then the organism will try to survive. When it will try to survive, it will produce something which is not required for its growth. It will actually help other organisms to die so that it can grow easily in that environment. So for secondary metabolite production, mostly metal ions are used, which are actually toxic for these organisms. Then sodium, potassium, phosphorus, these are used for maintaining the osmolality, cell porosity. So if you want enzyme, we have to incorporate these so that the enzymes are being secreted out of the cell. Then halides. Now, chlorine is majorly used. Say if your organism is a halophile and you want to grow it in your fermenter, you have to provide it chlorine ions, that is salt. You have to provide it salt. Otherwise, it won't be able to grow. Okay. Then buffer. So I have presented a small graph. The top graph is of dissolved oxygen. The middle one is OD, and the last one is the drop in pH. On the x-axis, we have time, that is the fermentation log hours, and on y-axis, these three OD, pH, and dissolved oxygen. So if you see the middle graph, as the time increases, the fermentation goes on, slowly the population of the organism increases, it stabilizes and then declines. When it is in the exponential phase, most of the carbon source will be utilized. If the carbon is utilized, then the primary metabolites will be formed. And majorly, every organism produces organic acid. Now, because of accumulation of this organic acid, there will be a sharp decrease in pH, which you can see in the bottom graph. There is a sharp decrease in pH, and when the carbon utilization is almost complete, I told you in previous slides that carbon is directly proportional to biomass. So see, where the biomass is maximum, at that time the pH has dropped and it is now slightly coming up. Now there is no nutrition available. Now it will shift to nitrogen consumption. Now it will consume nitrogen, it will break down the amino acids, and ammonia will be released. And because of increase in ammonia, the pH of the media will become alkaline. Again, you can correlate this with the increase in pH trend during the late log hours. Okay. So why buffer? We are adding buffers to control this pH. So this is a perfect profile, but if there is some anomaly, then we need to add buffering agents like carbonates and phosphates in the media. Sometimes sterile sulfuric acid, NaOH, or ammonia gas is also purged in the fermentation media to maintain the pH. Now, there are many fermentation processes where we need some precursor molecules. The organism is not able to produce that, but if you are giving them some precursor molecule, then they can convert it into the desired final product. This is also known as biotransformation. For example, L-tryptophan, which is an amino acid. So if you give L-tryptophan and if organism has an enzyme for the production of indole acetic acid, which is a plant hormone, so it can produce IAA, which can be used in any tissue culture or for the apical growth of the plant. Then monosodium glutamate. You must have used Ajinomoto in Chinese food. It is monosodium glutamate, which is food enhancer, taste enhancer, and this can be used for as a precursor molecule for polyglutamic acid, which is a very good moisture trapping agent and being used for cosmetic application. Now, inducer, they are the substrate analogs. Like I want to make amylase, so I will give maltodextrin instead of cellulose or starch, and my organism will start producing amylase. I have to give some now Piscia. Piscia pastoris is a expression. What do you say? I forgot the term. Like you have expression vectors. So when you when this expression vector is transformed inside Pistia pastoris, the Pistia pastoris promoter is getting activated by methanol. So what happens if the organism is provided a small shot of methanol, it will start producing and expressing the desired gene which has been cloned inside it. So methanol is an inducer. Next, please. Oxygen requirement, yes, it is most critical because without oxygen, at least the anaerobic fermentation cannot happen. So it needs to be controlled, and it is required for fast metabolism. For every single atom of nitrogen, 4 times more amount of oxygen is required than for a carbon source to be utilized. So it is 4 times more oxygen which is required for nitrogen degradation. Then oxygen requirement is also dependent upon rheology. Rheology means the viscosity, the fluid property of the fermentation broth. As I earlier mentioned, if it is more viscous, the oxygen penetration and staying inside the broth will be restricted. And antifoam, this also I have discussed earlier. Now the major thing which we work upon is media optimization. So this is what, these are earlier whatever we have discussed are the different components of the media. Now we come about different concentrations. So how we decide how much amount of particular component is required. So this will be a little little difficult to understand. So I have tried to make it very simple. There are two approaches. One is classical approach and one is factorial approach. What happens in classical approach? We take one variable. Say I told you 5-6 variable: carbon source, nitrogen source, oxygen requirement, maybe pH, temperature. These are the different variables which actually determine how much product will be formed or how efficiently my fermentation will happen. So if I want to optimize each and every parameter, so there is a formula like x to the power n, where n is the number of variable. Say I have 4 variable: carbon source, nitrogen source, pH and temperature. Then x is the concentration or the value, the levels at which I have to take. So for example, there are 6 nutrient components and 3 concentrations I want to take up to check whether our organism is able to produce maximum metabolite or maximum enzyme at which concentration. So I will have to keep 729 trials, 729 flasks. Is it experimentally feasible? No. So, and even if I try to do it, I may end up with huge amount of data, which is not easy to analyze. So it is very time consuming. I cannot handle large number of variables. Say it is just 6 and I am not able to handle these many trials. If it is 10, then how will I handle? Then the interdependency of the carbon and nitrogen source, that also I won't be able to check. So there comes a factorial design in which what happens, I have to take only two concentration, a lowest concentration and a highest concentration. And the number of trials will be number of variable plus 1. Say I have to check 4 variable, so I will keep only 5 experiments. And how I will decide? So there was a scientist named Plackett and Berman, and they have given this design. This is small table for different number of variable. They have defined different table. So first line of this table, if you see minutely, H and L are written, and on the left side we have 1, 2, 3, 4, 5, 6, 7, 8. So say I have 7 variables, I will keep these many, that is 8 different flasks, and in each flask the concentration of my components will be either high or either low. So these are the different compositions. So what happened at the end of the experiment, I will have different yields. That is the response. If you see in the right hand side, there are different yields. So these are the yields which are actually taken into consideration, and by using some mathematical statistical models, then we devise which concentration is actually required at an optimum level. So these are the two different methods which are majorly used in industry for optimization of process. I'm pretty sure that factorial design must have gone like a bouncer, but please go through this after my lecture, and you can Google it out. I'm pretty sure it is not that difficult. Next, please. Now comes the sterilization. I'm very much aware that you must have learned all the sterilization procedure. I will not go in detail. I have just made a flow chart, physical, chemical, and mechanical method. The most important and 90% used in industry is the sterilization process, where we use steam. Now, going a step back, there are different limits at which heat is being used for sterilization. And I won't say that any of these methods is of less importance, because in an industry, we need to have sterilization at each and every level. For example, in lab, the tables, benches need to be sterilized and disinfected. For that, we have different strategy. The glasswares need to be sterilized. For that, we use oven and different strategy. For hand sanitization, we use 90% alcohol. That is again a different strategy. The petri plates which we are using are gamma irradiated. So this is a different technique of sterilization. And then for the media, of course, we use moist heat. Then say if it is a milk producing or dairy industry, then it will use pasteurization rather than complete sterilization, because they don't want to kill everything. They want the probiotic organism, the lactobacillus, still in the product. So they will not kill everything. They will just reduce the load of the microorganism. So quickly we will go through 3 terms which are very important. One is sterilization, complete death of all the organism present in a sample. Disinfection, it is decrease in the amount of bacterial or pathogenic load. Okay, it is not complete killing of all the organism. Antibiotics, these are the substances which are given to target a particular group of organism. It will not kill all the organism. Okay, and then aseptic is sterilized environment. So if I say that as a personal, what would you prefer, a sterilized item or a sterile item to work with? Then you should always opt for a sterile item, because a substance can be sterilized, but over the time it can get contaminated. So a substance labeled as sterile means the sterility of the organism is maintained, and we should prefer such object to work with. Now, for heat sterilization, if the temperature is below 100 degree, that is pasteurization. Earlier, it was done at 65 degrees for 30 minutes, but nowadays, ultra high temperature, that is UHT systems are being used, where 72 degrees is maintained for a few seconds and the pasteurization process is over. So this is because this is for samples which is having very high protein content. So when the protein is subjected to high temperature, it may get denatured. So this is how we maintain the property of the product while we sterilize it to a limit. Then Tindalization, that is mostly for sugars, because in sugars, um, sugar is also a cryoprotective agent, and it can also restrict the steam to penetrate into the cells. So if a sugar solution is of very high concentration, the steam could not penetrate through it. So in that cases, it is tindalized, that is, it is boiled at 100 degree for certain time period. It is left for few hours so that any spore or any organism which is still there may grow, and then again it is subjected to boiling successively 2 or 3 times. So this is known as tindalization, which is done at 100 degree. When it is more than 100 degree, it is known as sterilization by autoclave, that is steam sterilization, and mostly it is done at 121 degree for 15 minutes. But depending upon del factor is the count. It is a factor which determines the count of organism at after sterilization, that microbial load after sterilization. So this del factor can be achieved for at different temperature and time regime. So say if I autoclave some substance at 121 degree for 15 minutes, it is equivalent if I autoclave the substance for 126 degrees for 10 minutes. So what happens? I could achieve the same del factor. That is, I can achieve the same killing of the cells, but the damage to the media will be less. Second, lastly is the dry heat, that is 160 degrees, and mostly glassware, syringes, metal items, surgical items, sometimes some powders, mixing powders, talcs. These are sterilized oils. These are sterilized for 2 hours in dry heat. Now, there are many substances which cannot withstand heat, and they are heat labile. So, for those substances, we perform filtration. So, for filtration, I have grouped them. What is the pore size? Like for precipitates and clots, it's 5 micron. For microbial, mostly 0.45. But if you want absolutely pure, that is like vaccine-like substances, so for that you need 0.1 or zero point 0.2 micron, because most of the microorganisms cannot pass through 0.45 micron. But there is a bacteria named Brevantimonas dysminuta that was earlier known as Pseudomonas dysminuta. So this is an organism which is 0.3 micron and could pass through 0.45 micron filters. So that is how 0.2 micron filter came into existence. And for filtering out mycoplasmas and endotoxin, 0.1 micron, and sometimes like in this pandemic situation, to filter out substances of viral load, we need 0.045 micron filters. Next, please. So quickly, I will just take 15 minutes more. So all the steps which I have discussed till now are being used in research and also in preliminary phase of fermentation. Now, what is scale up? Now, you have isolated an organism, characterized it well, identified it, preserved it. You have designed a media where it can grow happily and produce a desired product. Now what? Now, can I produce it in 100 ml and go and knock everybody's door and give that 100 ml fast? No. So, if I want to commercialize it, I need to produce it in a large scale. So, what is the process? So, when I am producing it at a very small scale, there are many aspects which I have to look into. So we will go in next slide. I will explain it I will explain it properly. Just to show you a pictorial representation in lab, what people do during research, they produce culture or produce their desired product at 100 to 1 liter scale. That is in flask. A maximum big flask is 2 liter, and you can accommodate around 1 liter of media in it. Now, if I want to scale up, that is go to a higher level, I have to increase the amount. So the amount can be increased, and there are small bioreactors available, benchtop bioreactors, which are 1 to 2 liters. Sometimes they are 10 to 50 liters also. So which are 10 to 50 liters, they are ground mounted. They are not on the benches. So they are not known as bench tops. Then I am successfully able to grow in that, but later I feel that the requirement is much higher and I want to again increase the volume. So I will go up to 1 kiloliter, that is 1000 liter. So again the amount of fermenter, the volume of the fermenter will increase. So with this increasing volume of the fermentation broth and the fermentation vessel, there will be many physicochemical properties which will change, because like the way the broth is getting mixed, the way oxygen is getting dissolved, the way nutrients are available, the shear, because of the vortexing, a shear strength is created inside the vessel, because of which cells may rupture. So these things cannot be studied properly in lab scale. So we have to take this slowly, and this is what scale up means. Next, please. So what is the purpose of scale up? This purpose is very simple. We have demonstrated, we have done few experiments at lab scale. Now we want to take it up to commercial scale. So we have to check it at every level. Where what we are getting, the results we are getting, the fermentation efficiency which we are getting at a small scale is getting replicated at large scale or not. Second, during this process, we will also be able to generate some data which will help us design a bigger fermenter or a bigger production facility. Third, it will also help us to generate a material which can be analyzed. Say, everybody must be having probiotic dahi, curd. So this, have you ever read the ingredients? It will have lactobacillus or bacillus coagulans present in that. So how does bacillus coagulans cause curdling? No, but it is added as a probiotic. So what people do, they produce bacillus coagulans in bulk, and then they mix small amount of this in powder form. They mix it in the milk, and then they allow the milk to curdle. So what happened? The product, that is the bacillus powder which is mixed, need to have some characteristic. If the powder which the company is producing, that is the bacillus cells which the company is producing, is black in color, so you will get a dahi which is black in color. Would it be acceptable to you? No. We want a clear white curd. So the quality, the physical appearance, the purity of the final product is actually more importance to the for the customer. So we during this upscale, we also produce the powder which can be assessed or can be studied for its property. What are the rules while doing the scale up? So first and foremost rule is we have to maintain the geometry and configuration of the fermenter. Say in lab scale, I am doing a fermentation in flask. In benchtop, I am using a normal fermenter, but while scaling up, I am using a airlift fermenter. Will it be possible? No, the geometry and configuration change entirely. So I have to use a same kind of fermentation vessel with a difference in volume, okay, for the scale up procedure. And this increment in volume should be always should be in magnitude of powers. It should not be like liter. Like pehle to main I was dealing with 1 liter, and then 1 liter 100 ml. So this is not scale up. when I am scaling up some process, I have to be in folds, like 10 folds, 50 folds, 100 folds. That fold increment is requirement. And any scale up cannot be in 1 or 2 steps. It should be a minimum of 3 to 4 steps. It can be 7 to 8, but a minimum of 3-4 steps of increment is required. So these are the 3 fundamental rules of scale up. Then what are the steps involved? I have to check. If I'm a industrialist, I have to check what is the manufacturing cost. Is my, during this scale up, is my lab results getting validated during the scale up procedure? I was getting protease, maximum protease production in a flask. But when I scaled up, there is something I'm getting 50% protease production. So that should not happen. And the scale up procedure, it's not like it won't happen. It may happen, but during the scale up, we have to optimize the process. And this is what process optimization also means. Then I have to check the control. There are a few controls like cleaning, sterilization. When we are sterilizing media in a small vessel, the steam going in, the temperature, the holding time will differ. So when it happens at a larger scale, there will be some different kinetics involved. So we have to study those things. Then how the product will be finally taken off. So the downstreaming also we can manipulate during the scale up procedure. Then what is the mass energy balance? Mass energy balance is the very critical part to determine a cost of a product. So the raw materials, how much raw material is I'm providing and how much material is getting converted into the end product. There is the statistics involved, which is actually responsible for costing a manufacturing procedure. Then there will be many system limitations. Sometimes dissolved oxygen could not be maintained. So those physical parameters need to be studied in detail during the scale-up procedure. Next. So once the scale-up is done, now we are ready with a performer, like with a sample procedure, how this process will work. Now, the main production is actually divided into 2 major parts. One is upstream and the other is downstream. Just for an example, say if I want to make curd in my home, what I will do, I will add a spoonful of inocula from a previous curd into a bowl of milk and then keep it overnight to get curd. But if I want to produce this, like Amul is producing this for so many people all over India. Will it be possible for it to do? No. With 1 spoon, they cannot make so much curd. So what they will do? So they have to increase the amount of inocula and they have to produce in bulk. So this whole process of like inoculum development and production is known as upscale or upstream. Okay, so where in the picture you can see there the first process is upstream, where from a flask we are inoculating the culture in shake flask in going in pre-seed, because you need a sufficient amount of cells actually for a bulk production. To reach that sufficient amount, you have to go through several steps, and these are the steps: shake flask, pre-seed, seed, and then the final fermentation vessel. So in that fermentation vessel, the culture will grow. If the biomass is required, then it will be harvested when the OD is highest. If the primary metabolite is required, then we will harvest in such a manner that when the enzyme or the primary metabolite are produced at a maximum level, and these all optimization has been done during scale up process. If a secondary metabolite, then we will wait for the stationary phase to reach, and when the secondary metabolite is produced at the maximum level, then we will harvest it. Okay, so the second phase is harvesting. So harvesting means what we need, we have to take it out. Okay, so we will end the process and just take out whole of the content. It is many a times it is considered within the upstream process. The major thing is the downstream, and it is very critical. Downstream is whatever we have produced, now we have to purify it and make it in a form which can be consumed by the consumers. So if it is a biomass, then we have to separate the cells, be it mycelial cells or be it spores or vegetative growth, whatever. We need to centrifuge it. There are many downstream processes like centrifugation or cell extraction. So those procedures can be applied depending upon your product. If it is a biomass, cells are taken. If it is a primary metabolite or secondary metabolite, the broth is taken into account. Say if it is a pharmaceutical company, we are more interested into a secondary metabolite. So what will happen first? They will take the broth. Now, when they are taking the broth, if it is an enzyme, enzymes are what? They are protein. So protein will be of particular weight. So they can filter it using ultrafiltration. So where the filtration membrane will restrict a particular Dalton or size of a molecule to pass through. So enzymes may get trapped in that and get concentrated, and then we take the retentate. That is the filtrate which is left is spent out, and the concentrated portion can be treated further for purification. If it is a secondary metabolites, first remove the unnecessary things. It can be precipitated out. So there can be different physical chemical methodologies, solvent extraction, chromatography, by which we can purify our final product. Now, final product can be either of liquid form, or it can be of solid form. If it is in liquid form, the shelf life may be less. If it is in solid form, the shelf life will be dry form, then the shelf life will be more. So we are bacillus manufacturing company. So we are producing only the bacillus cells. So these cells are then spray dried, and then the powder which we get, these can be sold off, and they can stay for 5 to 6 years also in a stretch. Sometimes when the organisms or the metabolites are heat labile, they cannot be spray dried, because in spray dried a very high temperature is required, around 150 degrees for a few seconds, but it is very high. So the cell damage or drying out can happen. So what people do, they adapt the method of lyophilization, but it is a cost intensive method. Always keep in mind, because when there is commercial entity involved, they are money minded, they think how to save money. So this is a precise way of how production happens. Next, please. So these are the different applications. I have tried to summarize. The organisms or metabolite can be probiotic. They can be used in cosmetics. They can be used in medicines, in food additives, in agriculture, in treating oil spillage or biomarker or molecular, any aspect. There can be n number of applications. So first of all, we have to decide which application we want to, and then we should initiate. Next, please. So just quickly click once more. Again. Yeah, again. Yes. So this is an overview of, no, please go back. Yeah, this is an overview of any company, how it works, any fermentation company, how it works. So they have a research and development team, they have a production team, and they have a quality assurance and control team. So the research team will actually isolate, preserve, and characterize the culture. Once they are done with that, they will also optimize the process. Then collectively with the production team, they will perform the upscaling. Then for the final production, from shake flask, seed fermenter, fermentation, the production team will be solely responsible for this process. And then either there will be a biomass produced, or the metabolites will be produced, which can be separated or concentrated, which is a downstream process, and then we get a final product. Now this final product, it should be up to the mark. That is, it is given to consumers, human beings or plants or animal. So they have some defined quality. The customer will have some defined quality, and some testing should be done on it. The qualities can be physical, that is, they should look like if you are adding something in food material, application in food material, it should not be foul smelling, it should not be black in color. So all those things matter: the appearance, the palatability, the free flowing nature. These are few physical characteristics which are looked upon. If it is being used by human beings, the critical characteristic also, critical parameter also increases. We have to look about limit testing. What is it? Limit testing is there any pathogen? What is the limit of the pathogen present? No, like air. Air is the whole process cannot happen in a controlled environment. So at some stage, the product is exposed to air or the environment, and it can lead to contamination of pathogens. So limit testing is done to test whether any pathogen is there or not, and what is the limit which has been given by the customer. So depending upon that, then there are compliance agencies like FDA, Food and Drug Administration, so FDA, then IFCO, FSI. So these are the certification units which have their own criteria. So before launching a product into the market, we have to decide and check our process and the product for its quality. And for all this, the quality assurance and control team is responsible. So quality control teams control how the process should be accurate and the product is of pure grade, and to maintain its quality. A quality assurance team is actually assuring all the compliances. So this is how any company, any production company works. Next, please. Next, please. Amina, am I audible?
Yes, ma'am. The next slide is
So,
on.
yeah. So this is the factory bird view. So the left hand side unit is our production plant, and right hand side unit is the research lab. And I cannot show you the insights of the plant, but this is how we work. So thank you all. I hope I could give an insight of how an industry works and how you should tame your mind. Because when I was a graduate in biotechnology, I used to think that we will work in RDNA technology and I will do so many things. But when I completed my master's and I was subjected to the real market, then I actually understood how microbiology, how basic science are actually involved, how biotechnology is, where it is actually involved in the commercial and the real world. So this is all from my side, and now it's open for question and answer round. And you are equally, I'm equally happy if anybody wants to put up their questions in chat.
Students can put their questions in the chat box.
I think they find it little boring or what?
Okay, Anjali ma'am, you want someone to read the question?
No, I can read. So I think I have one question. If something goes wrong in any one step, then how it is rectified? So I am not sure if I get this question right. Any one step means all the 6 steps which I have told. So there is always a possibility. Yes. Okay. So there is always a possibility, like during the upscale procedure, if anything goes wrong, then we have a chance to rectify it, to modify our process. But it is quite possible during the main fermentation, there are chances of contamination, there are failures, but at a very lower risk, at a very lower level. So it happens regularly, but at a very low scale. And during the upscale process or the scale up process, all these challenges are taken into account. So what could be the chance of contamination, that are all looked upon during that scale. And then media optimization, if something is not working, everything is looked upon during the scale up procedure. I hope I have answered. If not, you can repeat or elaborate on your question in the chat box. What are the different industries where microbiology students can work upon? Very nice question. So microbiology has access in all the industries. Name it and you will have it. It can be food industry. It can be even the microbial industry, because chemical industry. Any chemical which is being produced used anywhere, it needs to be quality tested, and this quality testing department will always have a microbiologist. Any medical microbiology related industry will have its microbiologist. You name any textile industry will also have microbiologists, because the textile, any material, cloth material which is being produced, it is treated everywhere the microbiologist can have their scope. So I feel microbiology is the safest field which can be looked upon. Even biotechnologists should not feel disheartened, but they need to work upon the basic sciences. Mostly they have the illusion of working with the molecular biology, but this does not happen in real life. So give emphasis on basic sciences. Then what if in the quality assurance step we find that the quality is not up to the mark? Then what is done? Good. So really, mostly this don't happen, because industries always test their process and they have different control parameters in place, checkpoints in place. But many a times there could be such possibilities. In that case, the same thing is communicated, and sometimes recall also happen. Recall means, say I find that my product is not up to the mark. I cannot sell it to the customer. It is not advisable to sell it to the customer. If it is like customer is able to purchase it, many a times we set a limit. Say I want 100% purity, but 80% purity is also good to go. So I will give a criteria to the production team that I need 100%, but in case they end up with 90% sterility, I can still launch the product. Okay, but many cases like which are very critical for food, for vaccine, for medical application, if the product is not passing the quality, they need to be discarded and rejected. So that is the take from commercial point of view. Are there any standards given to sustain the products before selling to the consumer? Yes, there are many standards, and these standards are defined by the government. Like I mentioned, there are many certifications. Even just for your knowledge, like industry should have good laboratory practices, good manufacturing practices. So these are the certifications which are given by different agencies and which are properly audited. There are different certification for food grade, different certification, other certification for application in pesticides. So name a few, like CIB. CIB is responsible for any agricultural registration, audits and registration. FISI is responsible for any certification or quality assurance for food products. So these are the different type of like HACCP auditing system is also there for food. There are different certifications, many more certifications. What is the actual work of a medical microbiologist in a hospital? So in a hospital, you go for testing. Like you are having some fever, which is there for 5-6 days. So doctor may ask you for going for a urine culture or some testing. So those testing in the labs are done by the medical microbiologist. The extent of infection, RT-PCR. So these days, people know the pandemic situation. So the medical microbiologists are in demand. So you have to culture the blood samples. You have to isolate the strain. You have to check how much mutation has happened. You have to devise a remedy for that. So there is the scope for the medical microbiologist. The extent of infection, everything. The product which is rejected, is it considered as a bio waste, and how it is disposed of? Very good question. For any commercial entity, when you are in academics, you have to just discard out your sample. So you can just decontaminate. That is, that you can sterilize it in autoclave and discard it or throw it in, or you may have to incinerate the product. But when it is a commercial entity, you cannot. There is proper pollution control board and proper effluent treatment board. You have to have a EIRA facility, that is environmental risk assessment facility, available in your compromises, which look into the standards which are being defined by the regulatory bodies. Okay, so for our company, we produce bacterial biomass. The effluent which we produce, spent biomass, is first treated in effluent treatment plant, where we provide more oxygen so that if there is an organism left, they can grow. Then it is heated at a high degree, sorry, and then it is being utilized for farming. That is, inside, I have shown you the image of our company. So we have quite a amount which is covered with green plantation. So we use that effluent in soil reclamation. So that is how we are maintaining our effluent, and it is a zero based management system we have in place. But for many other companies where they cannot treat it to zero waste, they have to deposit it to the pollution control board, or there are different agencies which are responsible for their treatment further. So you have to be bound with the compliance agency. Okay, thank you. I think I have answered all the queries. Still, if you have, I can free to answer. Yeah, we always study about the ethics in chapter of biotechnology. So isn't exploiting microbe against some ethical issue? No, not at all. But there are agencies like National Biodiversity Authority. So earlier what happened, I will give you a small example. In India, we used to consume turmeric in day out, and we used to think that it is enhancing our immunity. Later, one nice day, America decided to patent it as their product. So our patenting authorities then claimed and showed them evidences which dated back in historical era that turmeric is being used extensively in our household for the purpose of enhancing, boosting up our immunity, and then they have to withdraw it. Similarly, using a microorganism for growing something is the safest, for growing or exploiting them for commercial use is the safest way. If you are using an animal, it is an ethical issue, and we cannot use it unless there is an approval from the ethical committee. But for microorganisms, we can exploit them. The only thing is, if we are over exploiting a particular biome or an ecosystem, we have to address this to the national biodiversity. If we commercialize it, we have to address this to the National Biodiversity Authority. And while commercialization, we have to pay them an amount of royalty, which will be utilized for the conservation of that biome or that ecosystem. So that is how we are maintaining a balance between nature and commercialization. And this National Biodiversity Authority has now come up with many rules and regulations for commercialization of microbes. So it was really a very good question from the audience. How can a microbiology student get into R&D industry? Can a graduate enter? Mostly graduate cannot enter into R&D because research requires experience. So whatever you have learned, that is all bookish during your graduation. Unless and until you do masters, where you have, along with your coursework, you have a dissertation, where you have actual experience, hands-on working facility available. So you learn the thing, then only you can proceed with research. You need to have an analytical mind. So I won't dishearten or say that you cannot enter. There are few industries which encourage by graduate students to enter into industry, but mostly after masters, you need to have an in-depth knowledge of the subject. Only then you can enter into an R&D. Mostly R&D of industry need to be very skilled. Maybe PhD and postdocs are only taken for these positions.
Thank you, ma'am. It seems that all the questions have been answered. Thank you for that, ma'am. Now, I would like to call upon Ms. Priyanka Yadav to please deliver the formal vote of thanks for today's session.
Okay, before that, I would like to thank the organizing committee for giving me such an opportunity to actually interact with the students. Maybe we could offer some opportunity later when they are eligible for our criteria. So I am really grateful, and such type of interaction, I would encourage you all to plan a visit in our facility. Maybe because of this pandemic, it is not allowed right now, but later you are encouraged to have such interactions. Thank you.
Yes, ma'am.
Good evening, everyone. I, Priyanka Yadav, on behalf of KC College Department of Microbiology, deem it a great honor to propose a formal vote of thanks to all those who have helped us in making this session such a resounding success. First of all, I would like to propose a hearty vote of thanks to our very dynamic principal, Dr. Hemlata Bagla, Head Nuclear Radiochemistry Department, Senior Dean of Science and Technology, for awakening the passion and desire to support us as a ladder to reach the pole of success. Ma'am, your constant encouragement has always helped us in all possible ways. Thank you so much for always being there for us. I would also like to thank Dr. Sagarika Damle, DBT Star College Status Coordinator, for helping and supporting us in all our endeavors. I would also like to express my heartfelt gratitude to our distinguished speaker of the day, Dr. Anjali Bose, Head of Strain Management Development of Zytex Private Limited. Thank you so much, ma'am, for making our students aware of how an industry works, introducing them to biome, bioprospecting, what is R&D, upstream and downstream processing, applications of each step and its importance as well. Thank you for enlightening them with great ideas. Yes.
Thank you so much.
And thank you so much for patiently sorting out the queries put forth by the students. I would also like to take this opportunity to place on record my hearty thanks to the organizing team for being so proactive. Their meticulous work and dedication have made this event a success. I would also like to thank the people who have worked behind the scene to make this event happen. That is our technical team, especially Monish, for providing all the help and support. Last but not the least, a special thanks to all the audience for showing such a great response. Thanks for being here today. Thank you so much.